What if the deadliest weapon in the human immune system is actually the exact cure for cancer we've been looking for?
Right. It's this massive, completely counterintuitive biological paradox.
It really is. And that's exactly the dilemma we have for you today. You know, usually when we talk about getting a medical diagnosis, there's this expectation of precision. Like if you break your arm, the x-ray shows a jagged white line and the doctor just points at it and says, there it is, we fixed that.
Yeah, it's clean. The problem is incredibly localized and the intervention is straightforward.
Exactly. It's a mechanical problem with a mechanical solution. But then you step into the world of oncology and immunotherapy and suddenly that x-ray machine is basically useless. We're no longer looking at a broken bone. We're looking at a microscopic war zone where the enemy is like actively disguising itself as healthy tissue.
And that disguise is what makes cancer so notoriously difficult to treat.
Right. And that brings us to the core of today's deep dive. What actually happens when medical science discovers a biological weapon powerful enough to single-handedly win that microscopic war, but it is fundamentally just too dangerous to actually put inside a human being?
It creates the ultimate therapeutic catch-22, for researchers.
Yeah.
You have a potential cure sitting right there in a vial, but the collateral damage of using it is literally fatal to the patient.
And navigating that exact tension is what we are unpacking today. We have our hands on a really fascinating 2026 paper published in Cell Reports Medicine by Tang and colleagues. And they're detailing this massive breakthrough in cancer immunotherapy called the T12 fusion protein.
It's a remarkably elegant piece of bioengineering.
It really is. So our mission for this deep dive is to figure out exactly how these researchers took a toxic biological bomb, put a leash on it, and engineered it to detonate exclusively inside a tumor while, completely sparing the rest of the patient's body.
Yeah, and to really grasp the magnitude of what Tang's team accomplished here, we kind of have to look at the current landscape of cancer treatment for a second. Over the last decade or so, therapies that target T cells like PD-1 blockades have revolutionized how we treat cancer.
They've been game changers.
Absolutely, but they have a glaring blind spot. They routinely fail against what oncologists call immunologically cold tumors.
Right, cold tumors.
Yeah. These are cancers that essentially build a chemical fortress around themselves, making the immune system just completely ignore them. The reason this new paper is turning so many heads is because this T12 molecule successfully breaches that fortress.
And it doesn't just breach it quietly, right?
Not at all. It engages both the innate, rapid response immune system and the highly specialized adaptive immune system at the exact same time, basically turning those cold tumors entirely hot.
Okay, let's set up a bit of an analogy to carry us through this just to make it easier to visualize. If your immune system is a car, traditional cancer therapies generally do one of two things. They either press down really hard on the gas pedal to speed up the immune response, or they cut the brakes that the tumor's using to slow the immune system down.
Right, gas or brakes, usually not both.
Exactly. But today, we're looking at a molecule that manages to do both simultaneously. But, and here's the genius part we're going to explore, it only performs this maneuver when it detects that it is parked securely inside a tumor.
That car analogy works perfectly for the mechanics here, I think. But to understand why T12 is such a necessary leap forward, we really have to talk about the flawed superstar it was built to replace.
The dangerous weapon we mentioned earlier.
That's the one. That superstar is a naturally occurring cytokine called interleukin-12, or just IL-12 for short.
The biological weapon that's too dangerous to use.
Exactly. So think of a cytokine as a biochemical text message that immune cells use to communicate with each other. Wild-type IL-12, meaning the natural version that your body makes, is essentially a massive blaring megaphone.
Just shouting at the immune system?
Literally, when it's released into the bloodstream, it violently wakes up natural killer cells, which are your innate immune system's first responders, and CD8 plus T cells, which are the specialized assassins. It just screams at them to proliferate, attack, and destroy. And in a petri dish, it is incredibly potent at driving an anti-tumor response.
So wait, if IL-12 is this incredible immune megaphone, if it's so exceptionally good at melting cancer cells in the lab, how come it isn't, the standard of care in every single hospital right now?
Well, because of what happens when you hand a megaphone that loud to the entire circulatory system.
Yeah.
It doesn't just wake up the immune cells stationed specifically near the cancer, it wakes them up everywhere, all at once.
Oh, wow, okay, so it just floods the system.
Right. When oncologists tried injecting wild-type IL-12 into patients during early clinical trials back in the day, it activated the immune system so aggressively across the entire body that it caused severe, life-threatening systemic toxicity.
So it's basically a systemic carpet bomb.
It is exactly a carpet bomb. It triggers what we call a fatal cytokine storm. The immune system goes into such a complete frenzy that it essentially starts attacking the patient's own healthy organs, the liver, the lungs, the kidneys.
Which is terrifying.
Truly. So the clinical application of wild-type IL-12 was completely paralyzed by this toxicity for decades. The field knew how powerful it was, but they just couldn't deliver it safely.
Okay, so if IL-12 is essentially biological poison when given systemically, how do you put a leash on? I mean, how did Tang's team actually manage to confine this carpet bomb strictly to the tumor?
They solved it through some really clever molecular engineering. They created what's known as an immunocytokine. They took that powerful IL-12 cytokine and physically fused it to a tiny, highly specific piece of an antibody.
Wait, just a piece of an antibody?
Yeah, they didn't use a whole bulky antibody. They just used the single-chain variable fragment, what we call the scFv. You can think of the scFv as just the claw of the antibody, the part that actually grabs the target.
Oh, okay, the claw. I like that.
Right. And they used the claw of an antibody called 13G6. This specific claw has one single job. It intensely seeks out and binds to a cellular receptor called TIGIT.
Okay, I have to push back here for a second, because fusing a massive volatile cytokine to a tiny antibody fragment sounds, well, incredibly unstable. Doesn't this giant Franken molecule just sort of fall apart in the turbulent environment of the human bloodstream before it even reaches the tumor?
That's a really fair question, and it's the historical challenge with fusion proteins. But scFv fragments are actually engineered for incredible stability. Because it's a single continuous protein chain rather than a complex multi-part structure, it folds very, very tightly.
So it's structurally sound.
Very much so. It acts as a highly stable chassis to carry the IL-12 payload through the bloodstream completely intact. But honestly, the real elegance of the design isn't just the stability. It's the target they chose. TIGIT.
Right. Why TIGIT? Out of all the receptors in the human body, why did they choose that specific target as their homing beacon?
Because of what TIGIT does, and more importantly, where the tumor actively forces it to be. TIGIT is an inhibitory receptor. So going back to your car analogy earlier, TIGIT is literally a parking brake on the surface of immune cells.
A parking brake, okay.
Yeah. When a T cell enters a tumor, the cancer cells constantly bombard it with signals, forcing the T cell to stay continuously active. Over time, this chronic exposure causes the T cell to become totally exhausted. As it gets exhausted, it starts plastering its own surface with these TIGIT parking brakes.
Wait, let me make sure I'm getting this. So the tumor is intentionally forcing these immune cells to express TIGIT, slamming on their brakes so they can't attack the cancer. And Tang's team realized, hold on, if the tumor is filled with these specific brake pedals, we can use that exact defense mechanism as our homing beacon.
That is exactly the brilliant inversion of this therapy. By fusing the toxic IL-12 to the anti-TIGIT antibody claw, they created a drug that completely ignores healthy, circulating immune cells.
Because healthy cells aren't expressing high levels of TIGIT.
Spot on. Instead, the drug zeroes in directly on the exhausted, heavily braked immune cells that are trapped deep inside the tumor microenvironment.
That is so clever. And the researchers have the biodistribution data to actually back this up, right? It's one thing to draw this up on a whiteboard, but they actually proved it in living animals.
They absolutely did.
Yeah. The team injected fluorescently labeled T12 into mice that had MC38 tumors, and after 12 hours, they looked at where the drug actually ended up. T12 showed a 65.8-fold enrichment in the tumors compared to the surrounding healthy tissues.
Which is a massive concentration.
Right. Meanwhile, the old wild-type IL-12, it only had a tiny 2.6-fold increase. The old drug was just washing uselessly and toxically through the whole body, while T12 was locking directly onto the target.
Yeah, that 65.8-fold enrichment is the definitive proof of concept here. The anti-TIGIT antibody claw is successfully grabbing onto the tumor-infiltrating immune cells, pulling the heavy IL-12 payload straight into the battle zone and heavily anchoring it there.
Okay, so let's just do a quick mini recap for you listening to make sure we're tracking the biological mechanics here. IL-12 is powerful but too toxic for systemic use. TIGIT is an inhibitory receptor, a brake on exhausted immune cells inside tumors. And T12 physically links IL-12 to a TIGIT-targeting antibody, ensuring the payload only detonates inside the tumor.
That's a perfect summary. It's a literal lock and key mechanism that ensures the bomb only goes off once it's inside the castle walls.
Which naturally leads us to ask, well, what happens once the drug is inside the castle? Navigating into the tumor is a massive achievement on its own, but you still have to wake up the immune cells that the tumor has basically put into a coma.
Right. How does T12 actually remodel that hostile environment once it gets there?
Because if the guards are deeply unconscious, just standing next to them with a weapon doesn't exactly win the battle.
No, it doesn't. And these immune cells are as unconscious as they get. To figure this out, the researchers used a technique called flow cytometry, which is this incredible high-speed laser-based machine.
How does that work exactly?
Well, it sorts and analyzes the physical properties of thousands of cells per second. It basically allows scientists to look at the exact status flags on a single cell's surface. And they saw that T12 wasn't just binding randomly inside the tumor.
It was being selective.
Highly selective. It preferentially sought out the absolute most terminally exhausted CD8 plus T cells. Specifically, cells throwing up multiple flags of defeat, like the markers PD1 and TIM3. These are the soldiers that have completely given up the fight.
Okay, for you listening, I have a memory hook to help visualize what happens the millisecond this T12 molecule binds to those defeated cells. Let's call it the T12 two-step.
I like that. Let's hear it.
Okay, so step 1. The antibody claw part of T12 binds to the TIGIT receptor, which physically blocks the receptor from receiving any more suppressive signals from the tumor. That effectively wedges the break open. Step 2. Now that the molecule is physically attached to the cell, the IL-12 part of the fusion protein delivers a massive point-blank stimulatory signal. That slams on the gas. So wedge the break open, slam the gas.
The T12 two-step, that's highly accurate. And the resulting biological explosion from that two-step is just staggering, especially when we look at the innate immune system, specifically the natural killer cells.
Right.
To understand this part, the researchers utilized single-cell RNA sequencing, or cRNA-seq for short.
Just briefly, because we throw these terms around, what is single-cell RNA sequencing actually letting them see that the laser flow cytometry couldn't?
Good question. Think of it like reading the barcode of a cell's current mood and activity level. Instead of just looking at the structural flags on the outside of the cell, cRNA-seq lets you look inside every single individual cell to see exactly which genes are actively turned on or off at that exact moment in time.
Oh, wow. So you got a real-time look at their internal machinery.
Exactly. And what they saw was profound. T12 pushed the intratumoral NK cells to aggressively proliferate. They saw a massive spike in a protein called Ki-67.
And Ki-67 is a marker for what?
It's a classic biological marker that a cell is actively dividing and multiplying.
Okay, so they didn't just wake up. They started cloning themselves into a whole army. But what are they actually doing to the cancer once they multiply?
They are actively melting it. T12 induced these newly multiplied NK cells to secrete incredibly high levels of immune-activating proteins, primarily IFN-I and TNFN.
Which are basically chemical weapons.
Yes. You can think of these proteins as a combination of biological acid and a homing beacon. They directly damage the tumor cells, literally forcing them to break apart, while simultaneously altering the local environment to attract even more immune cells to the site.
That's incredible.
It is. The single-cell data showed the NK cell population actively shifting into highly aggressive cytotoxic, meaning cell-killing, and chemotactic, meaning able to attract reinforcement states.
So that perfectly covers the rapid response innate system. But what about the adaptive side of the immune system, the CD8 plus T cells? Because usually, standard therapies only successfully target one or the other. You rarely get a coordinated attack.
And that bridging of the gap is where T12 shows its true potential. For the CD8 plus T cells, T12 effectively reversed their terminal exhaustion. It fundamentally reprogrammed them. The cRNA-seq data showed a massive expansion of what we call early effector-like and progenitor-like T cell populations.
Let's translate that for a second. What does a progenitor-like T cell actually mean for the patient?
It means stem cell-like endurance. Essentially, T12 takes a dying, exhausted T cell that has absolutely no fight left in it and reverts it back into a fresh, aggressive, highly capable soldier that is ready to divide and sustain a very long-term attack.
It's basically an anti-aging serum for exhausted immune cells.
That's a great way to put it. But here's the most crucial detail of all this. They aren't just fighting as isolated units.
Right, because immunity isn't just about single cells firing blindly. It's about network communication. And this paper highlights a really nuanced crosstalk mechanism, doesn't it?
It does. The researchers meticulously mapped out the ligand-receptor interactions inside the tumor. Think of these as the biochemical text messages sent between different cell types.
Okay.
They discovered that T12 profoundly enhanced the communication network between three distinct groups: innate dendritic cells, natural killer cells, and the adaptive CD8 plus T cells.
The dendritic cells are essentially the intelligence officers of the immune system, right?
They are the scouts and the commanders.
Yeah.
Dendritic cells consume pieces of dead tumor cells, process those antigens, and then physically present them to the T cells, giving them the exact chemical mugshot of what to kill.
Wow.
So T12 creates this coordinated multicellular siege. It wakes up the NK cells for the immediate innate attack, it rejuvenates the T cells for the sustained adaptive attack, and it boosts the dendritic cells to ensure the intelligence keeps flowing continuously between the two sides.
Okay, let's do another mini recap here. If we step back and synthesize this microscopic remodeling, the drug targets the most exhausted cells, cuts their TIGIT brake, hits them with the IL-12 gas, and forces the innate and adaptive immune systems to talk to each other to coordinate an assault.
Exactly.
That is just a beautiful biological mechanism, but we have to transition to the ultimate question for any medical researcher. Mechanisms in a Petri dish or a mouse cell assay are fascinating, but how does this translate to the macro level? Does it actually keep the host alive?
Right, because that is the only metric that truly matters at the end of the day. And to answer that, we have to start with the safety data.
Because we have to remember, wild-type IL-12 was completely abandoned in the 90s because it was a lethal carpet bomb.
Exactly. And the safety data in this paper is arguably the most dramatic vindication of their entire design. They ejected mice over a 2-month period. When they gave mice the old wild-type IL-12, a microscopic dose of just 0.0625 milligrams per kilogram caused severe, rapid weight loss and 100% mortality.
Wait, 100% mortality from just a fraction of a milligram?
Yes. Every single mouse died from systemic toxicity.
That is incredibly bleak. So what happened when they used the engineered T12?
The researchers kept escalating the dose of T12, basically looking for the toxicity ceiling, trying to find where it becomes dangerous. And T12 was perfectly tolerated with 0 weight loss and 0 mortality, even up to 2 milligrams per kilogram.
Let's just think about what that means for a second. That is roughly a 100-fold increase in the maximum tolerated dose. They are delivering 100 times more of this profoundly potent drug entirely safely because the SCF V-claw is ensuring it only detonates inside the tumor.
It's an unprecedented safety margin.
It is, but I have to ask the skeptic's question here. If it's that much safer, did they accidentally engineer the efficacy right out of it? Sometimes when you put a heavy loosh on a bomb, you muffle the explosion a bit too much.
Which is a very necessary question.
Oh.
To prove the drug still had its teeth, the researchers pitted T12 against the absolute hardest test in modern oncology: PD-1-resistant, immunologically cold tumors.
Okay.
They used 3 very specific animal models: B16 melanoma, E0771 breast cancer, and LLC lung carcinoma.
And just for some context for our listeners, why are those specific models considered the ultimate test?
Because they perfectly mimic the most notoriously aggressive, stubborn cancers we see in human patients. Things like triple-negative breast cancer. These are the kind of cancers that completely laugh at traditional checkpoint inhibitors.
They just shrug them off.
Completely. In their control groups, the researchers administered standard alpha-PD-1 therapy, which is a current standard of care in clinics right now. And it basically did nothing. The cold tumors continued to grow uncontrollably, completely ignoring the therapy.
But when they swapped out the standard therapy and administered T12?
It achieved complete tumor regression. The tumor shrank down to totally undetectable levels. T12 completely overcame the checkpoint blockade resistance by forcing the cold tumor to become hot.
Complete regression of a cold tumor is literally the holy grail for oncologists. But there's a darker clinical reality here we should touch on. Cancer rarely kills a patient through just the primary tumor, right? Like a breast tumor itself isn't usually what ends a life.
Unfortunately, no, it's not.
It kills you through metastasis, when it sheds cells into the bloodstream that travel and seed themselves in vital organs like the lungs or the brain. How did T12 handle the spread?
This is where the researchers designed a highly aggressive pulmonary metastases model to test systemic protection. They didn't just let a tumor grow naturally. They literally injected MC38 and B16 cancer cells directly into the tail veins of the mice.
Oh, wow. Why straight into the vein?
Because this perfectly simulates late-stage metastatic cancer, rapidly seeding the lungs with hundreds of aggressive tumor nodules.
The stakes really don't get much higher than that in an animal model, so what were the results there?
T12 effectively suppressed the metastatic lesions across the board. When they examined the lungs of the T12-treated mice, they saw a drastic sweeping reduction in tumor nodules compared to the controls.
That's amazing.
And furthermore, the overall survival of those mice was significantly prolonged. T12 didn't just clear the primary tumor site, it actually empowered the immune system to actively hunt down the metastatic spread in the lungs.
But the researchers did one last incredibly elegant experiment to definitively prove how the drug was doing this, right? Because in biology, correlation isn't necessarily causation.
Right, you always have to prove the mechanism.
Yeah, how do we know it's actually this combined network of NK cells and CD8 plus T cells doing the heavy lifting and not just some other random effect?
They ran what's called a dependency test. If you want to prove a specific cell is doing a job, you chemically remove that specific cell from the environment and see if the job still gets done.
Like taking a gear out of a clock to see if it still ticks?
Exactly. So they use specific depletion antibodies to wipe out just the natural killer cells in one group of mice and just the CD8 plus T cells in another group. Then they administered T12 to both groups.
And the therapeutic effect completely vanished. T12 stopped working?
Entirely. This is the definitive mechanistic proof of the paper. T12's magic relies entirely on the combined force of both the innate and adaptive arms of the immune system.
They need each other.
They do. Activating just one arm isn't enough to clear these deeply entrenched, aggressive tumors. You fundamentally require the synergistic crosstalk, that multicellular siege that T12 uniquely forces to existence.
Okay, time for our final mini recap before we wrap up. So T12 is roughly 100 times safer than wild-type IL-12. It successfully cured multiple types of cold tumors that are completely resistant to standard immunotherapy. And it acts powerfully against both primary tumors and distant lung metastases, all by requiring the innate and adaptive immune systems to work together.
That perfectly encapsulates the data.
So if we step all the way back and look at the vast landscape of oncology, what is the core takeaway for you? What does this paper represent for the future of how we treat disease?
For me, it represents a total paradigm shift in molecular drug design. The major limitation of early immunotherapies was their bluntness. They were basically sledgehammers that shattered the patient along with the disease. But by taking the time to physically fuse a profoundly powerful, highly dangerous cytokine to a hyper-specific checkpoint inhibitor, these scientists have engineered a true precision weapon.
A smart bomb.
Yes. They've overcome the toxic limitations of the past by using the tumor's own defense mechanisms against it, forcing the innate and adaptive immune systems into this incredibly localized, hyper-aggressive alliance.
It really is a masterpiece of biological judo, isn't it? It takes the tumor's own suppressive parking brake TIGIT and turns it into the exact homing beacon that delivers the tumor's absolute destruction. It is a beautiful example of utilizing the body's own biology, both its specific targeting systems and its destructive capabilities, just in perfect harmony.
The engineering is truly stunning, and I think it proves that sometimes the most potent weapon is the one we already have. We just needed a better map to deliver it.
And that leaves you listening with a really provocative thought to mull over as we wrap up today. We almost always think about the future of medicine being entirely new inventions, right? New chemicals, new synthetics.
Always chasing the new thing.
Exactly. But if we can successfully take IL-12, a molecule, we literally abandoned to the scrap heap decades ago because it was too wildly toxic, and successfully fuse it to an antibody claw to safely cure cold tumors, what other incredibly potent, previously abandoned molecular weapons might be sitting right now in our medical archives, just gathering dust, waiting for the exact right molecular delivery system to bring them safely back to the front lines?
It definitely forces you to rethink what a failed drug actually is.
It really does. The diagnostic muddy waters are definitely clearing up. Thanks for joining us on this deep dive into the fascinating world of P12. We'll see you next time.